Welcome to Biology Made Easy. Today we are going to discuss two techniques of drawing plant tissues. First technique is how to make low power optical microscope drawings or tissue map or low power plan of plant tissues. The second technique is how to draw high power plant tissues. Welcome. So first of all, let's look at low power plan or also known as tissue map. Now, in this one, you have these tissues, plant sections. All you need to do is to map up the areas of various tissue. Low power, sometimes the individual cells cannot be seen. So I have made this one for us to use as a demonstration. This is a transverse section of a monocot stem. Now, these areas form one tissue. All right. In these ones, you don't need to draw in the individual cells. So, what is a tissue? A tissue is a group of similar cells that perform the same function. So this vascular bundle, this area here is one tissue. That's the sclerenchyma. Another area here is another tissue, phloem, another tissue, cambium, and another tissue, xylem. So in drawing a tissue map, all that one needs to do is to map up the tissues. So in this one, if we are mapping up tissues, all we need to do is to draw the outlines, put in the outlines, and put in the various tissues, taking proportions into consideration. So here we have one tissue here, that's the sclerenchyma. We have the phloem, that's that tissue. We have the cambium, that tissue, and we have the xylem. What we want to say is that we come back to this, which I've already made. This one forms one tissue. You can't, even if you can't see the cells well, you see that they are a dark area, and that represents sclerenchyma, forming one tissue. So those tissues have been drawn in. If you look at this one, if you look at this, that seem to form one area. There is this that forms a tissue. There's this area that forms one clear tissue. And if you look at this tissue in the middle very well, you see that this area is another tissue. This area in the middle, these areas, you can see very clearly. So this forms one tissue, and here another tissue. Going back to look at it very carefully, this, which forms one tissue, is phloem. This one is xylem. It forms another tissue. One does not need to put in the various holes, that is the vessels. All we need to do is to map up. This is one tissue. This is one tissue, similar. This is one tissue. This is one tissue. This forms a big tissue. So this is what we made up here. This big tissue here is the ground tissue, which is parenchyma. Now we work out and come this way. This one is sclerenchyma. So we have it here, sclerenchyma, xylem, phloem. In tissue map, just map out the various tissues. Now we want to look at how we are going to draw our cells. Tissue plan, tissue map, low power optical microscope drawing of this. Because of these vascular bundles that are arranged in a ring in the center with a big pith here. If you couldn't make it out, you have to check my lesson on plant tissues to find out how you work out monocot stem, monocot root, dicot, etc. So in drawing this, we look at this structure very well. You can see a very thin layer of epidermis. Then you have a layer of cortex. Then you have endodermis with the thickening here of suberin thickening. And then you have this layer, which is pericycle. Very thick layer, many cells thick. But these two layers of cells, also of pericycle, looks different. So we'll give this another tissue. This and this and these are xylem. All right, so we'll give them a tissue, and here is the phloem. Look at this area of the xylem, thickenings like this, and then the pith. These are the tissues we are going to put in. So the epidermis is very thin. We won't be able to draw all the cells. We are only drawing some. So we go like this, and the epidermis is very thin. So we are putting in our thin epidermis. We are also putting in the cortex, taking proportions into consideration, and the endodermis is thicker than the epidermis, so we give it a bigger layer. So that's the endodermis, and then the pericycle. Now the pericycle here is all this layer, but it looks like these thin layers here look different. So we'll give it the different space, and then the pericycle is as big as the cortex, as you can see. Then those two very thin layers. Then we are going to put in the vascular region. Here we have these cells here, this tissue here. See this tissue? Another one is quite close to it. We have another one quite close to it. They are quite closely arranged. But looking at them very carefully, the outer layers are thicker. So they form a tissue, thicker. And then, if we look at it very carefully, we will see that this region here, look at it, they are also thickening. And in between these thickening, there's this lighter areas, which are the phloem. So we have a thickening here, and in between the thickening, you have those lighter areas. So anytime you work out what is happening well, a tissue map can easily be done. So this is it. And then a big pith. So one can continue to make it big or just draw a portion if you can. So the labels, the epidermis is here. Epidermis, cortex, endodermis, pericycle, another layer out there, pericycle. Then here we have the thick bit is sclerenchyma, the inner bit is the xylem, and here is the phloem. And then you have some sclerenchyma around here too, and then we have the pith. So low power plan or tissue map or low power drawing of TS of monocotyledonous roots. Now we want to shift our attention to look at high power microscope drawings. I want to demonstrate how plant cells can be drawn from high power microscope. If this is one plant cell, this cell has a cell wall and a cell membrane. So we put in our cell wall, and if there's another plant cell around, we'll have that plant cell also here. And this plant cell also has a cell wall and a cell membrane. Good. Plant cells, you always have the plant cell, and the cell has its cell membrane and a cell wall. And the other one also has its cell membrane and a cell wall. And so in plant cell, you have always a middle lamella, which is this one. So that is the middle lamella. And plant cells, usually there are other cells around. So we put in the markings for the other cells. There are other cells around. Good. So in drawing close-fitting cells, this is the technique. Always draw the outer wall first, outer walls first, outer walls first, outer walls first. That's the first thing. Then the second thing is to fit in the inner. That is close-fitting cells. Plant cells from high power, we draw in a few cells, 3, 4, 5 cells. So we'll draw 5 cells of this epidermis, and we'll start from the cell. So we put in the cell, and then that cell, and then this cell, outer walls first, always outer walls first. Outer walls first. And then, because there are other cells around, we don't leave the place open. We are drawing the epidermis. Now, after the outer walls, now we are going to put in the inner walls. And then they are very thin, so they are very close inner walls, very close, very close. Always outer walls first, then the inner walls. Because there are other walls, other cells around, they are not left open. So that is the epidermis. So we put in our title, TS, epidermal cells of a leaf. Now we want to look at the cambial cells here, the cambium. They are also close-fitting cells. Is here. This cell is also here. So we always put in the outer walls first and then the inner walls. No matter how thin they are, the inner walls should come. So the inner walls. I can see something like a nucleus in there. So I put it in, and the cytoplasm area, if it can be seen. So let me enlarge the cell. Enlarge the cell. always the outer wall first, and there are other walls around, cells around, so they are in. If the cells that I'm drawing, I could see the nucleus and vacuoles, I would put them in, but they are not evident in the micrographs that I am using. But I see one that's something that looks like a nucleus in one of them. So that is the nucleus in there, nucleus in there, nucleus. And you know, the cambium, the cytoplasm fills the whole cell. All right, it's filled with cytoplasm. Now, if we have a cell like the epidermal cells that we can see and can see the cells with cytoplasm inside them, then the cytoplasm and the nucleus should also be put in, but always outer walls first, inner walls in. So we put in all the inner walls very fast. The epidermal cells, if we can see the nucleus and the cytoplasm, of course, there will be a large vacuole in there. So you have vacuoles. Vacuoles are in there. And then nucleus, the nucleus should not be shaded. Nothing should be shaded in here. There's no shading. So we have the cell wall, we have the cell membrane, we have cytoplasm, we have nucleus, we have vacuole. Now we are going to look at high power drawings of cells that have intercellular spaces like parenchyma cells. But I want to demonstrate first how to make them, all right? So let's say you have a cell like this. And this cell, as we say, we always draw the outer walls first. So I have these two cells and the outer walls. And let's say there's an airspace here. So I put in the airspace before I put in the outer walls in this area. You put in the airspace. Okay, so there are other cells around. So I put in here them. And then I come to put in the inner walls. So the inner wall here, inner wall here, and the inner wall should not disturb the airspace. So the airspace is there, and I have the inner wall. The inner wall should have nothing to do with the airspace. So the airspace is there, and the inner wall is there. So here are the cells. And the airspace. So here is the airspace. Neat airspace. So that is the technique of drawing parenchyma cells. So let us draw the parenchyma cells of this root. These are root cortical cells, all right? And if you look closely at this, you can see there's an airspace here, another airspace here, another airspace here, small airspace, 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 airspace. So we are going to make high power drawings to show these cortical cells and their airspaces. So I want to put in the cells, draw the outer walls of the cell first. drawing the cell. There's a airspace here. There's a cell here. It also has an airspace here. Then there's the cell that goes round like this, which also has an airspace here. And this cell, which goes like this. And a cell also around it here. Cell around it here. It's a big kind of cell here. This one. And this big filling here. And another cell here. Small cell here. And there's a cell here. There's a cellular space here. And cell here. And this cell here. Just the cellular space here. And this cell goes like this here. And there's a space, the cellular space here. And there's a cell here. So we have these cells. These are the intercellular spaces. We don't disturb them. We now put in the inner walls. The inner walls, leaving out the intercellular spaces. So their inner walls are put in, not touching the intercellular spaces. So the inner walls are there, and we don't touch the intercellular spaces. Seem to be two cells joined there. So we put in the inner walls. And so we have other cells around, and so this one, you have this here, you have other cells around. Other cells around. We have an intercellular space here. And cells here. So the other cells around, they should also have so if we can see here, we see some grains, starch grains inside them. So we put in a few starch grains, which can be seen in the cells. So we have a few starch grains that can be seen in the cells. And we put on our labels. So we have the labels of our drawing. We have an intercellular space here. We have the cell wall here. We have the cell membrane. We have some starch grains. We have another intercellular space here. Intercellular spaces. So these are parenchyma tissue. So title, high power drawing of TS of parenchyma cells, that's the cortex of dicotyledonous roots. This brings us to the end of today's lesson. In our next lesson, we will look at how to draw parenchyma and sclerenchyma tissues and the high-power optical microscope.